Welcome everyone to this SDSC webinar. My name is Manuel Schröter. I'm a neuroscientist at the lab of Professor Hirlemann at the Department of Biosystems Science and Engineering at ETH Zurich in Basel. In the next 15 to 20 minutes, I will give you an overview on our project and the proposal that we sent in for the third call of collaborative projects by the SDSC in 2019. The title of our project was DeepEphys, using machine learning for biomarker discovery in human iPSC neuronal networks. Before we delve into the proposal and some preliminary results that I will share with you, I would like to briefly introduce our team. Our team is composed of PhD students and postdocs from a quite broad background, including bioinformatics, biomedical sciences, physics, neurosciences, and statistics. Hence, our project is a highly interdisciplinary effort. Here at BSSC, mainly two labs contributed to the project, namely the Bioengineering Laboratory of Professor Hirlemann and the Machine Learning and Computational Biology Lab of Professor Bock. From the SDSC, we were grateful to receive valuable support from Dr. Christian Donner, and I will highlight some of the work that he did later during the talk. Just briefly, the funds that we received by the SDSC went into financing the position of one PhD student, as well as computing resources at the High Performance Cluster Euler. When you ask people to explain what neuronal networks are, they might naturally think of and refer to the brain, and perhaps spectacular images like this one by the artist and neuroscientist Greg Dunn. And rightly so. The human brain is a fantastically complex organ comprising about 100 billion neurons and 1,000 trillion synaptic connections. Yet, a lot of the fundamental insight that describes how neurons and neuronal networks function have not been inferred from intact human brains, but much more simplistic models. I therefore hope that you bear with me if I now introduce to you the model system and workhorse of our SDSC project. What we will see in this time-lapse movie are 2D neuronal networks imaged with a light microscope over 28 days in vitro. As you can see, they start as single dissociated neurons and then form, as time goes on, new connections with other neurons. Such model systems have been heavily used for several decades and allowed to gain a lot of the knowledge that we have so far on neuronal physiology, such as axon outgrowth and ion channel dynamics. Having said that, it is also important to keep in mind that this is a model system with advantages, but also its limitations. Although optical means, such as light microscopy, is generally a good first step for the study of neurons, it will tell you little about how neurons actually communicate with each other. This is why we and others have been developing and applying methods to interface neurons with highly sensitive microelectrode arrays, or HDMIRs, that are able to capture with very high temporal and spatial resolution when and where a neuron fires an action potential. An action potential, or spike in simple terms, is the currency of how neurons send signals between each other and occurs when the membrane potential of a neuron is depolarized. High-density microelectrode arrays, and you can see an example on the slide on the left, are recording devices that come with several hundreds to thousands of readout channels and that can record from single neurons as well as from entire neuronal networks. As depicted in the EM picture, neurons do grow happily on mias, but there are many other neuronal tissues that have been studied on mias, including organotypic slices, spheroids, organoids, and other cell types. On this slide, I would like to give you some examples and a taste of what kind of readouts you might get from HDMIR recordings. On the top left, you see the ongoing spontaneous activity of an entire neuronal culture. Each blue spot here indicates the location of a neuron and a fired action potential. Yellow, on the other hand, indicates positive signal amplitudes likely generated from axons. As a neuron forms connections during development, the observed activity also changes. What we see here is the initially uncorrelated spiking activity of neurons during early development. That then gets replaced later on by population bursts during later stages of development. This activity is much more correlated. Finally, MIAs also allow the study of cellular and subcellular aspects of neuron, as depicted here on the right. In this clip, we see how a neuron and its neuronal footprint is distributed across the HD MIA. See, there's a lot of detail possible. For our SDSC project, we focused on HD MIA recordings from human neurons generated through cellular reprogramming from induced pluripotent stem cells, short iPSCs. In a nutshell, the idea was to use recordings either from healthy control or from a line with a genetically engineered disease mutation, and to look for mutation-specific electrophysiological phenotypes that downstream could eventually be used to screen for disease-modifying compounds. In our case, we used two isogenic cell lines of purified dopaminergic neurons that were generated using a well-established protocol, the protocol by Crick's et al. In addition to healthy controlled dopaminergic neurons, we used a line with a mutation in the SNCA gene A53T associated with early onset Parkinson's disease. Here's a brief overview on the main outcomes and milestones that we defined in our SDSC project proposal. First, we would develop a reproducible and scalable analysis pipeline for HD MIA recordings from human iPSC-derived neurons, harnessing the rich data that comes with these recordings. The pipeline would start with pre-processing of the MIA data, a computationally very heavy process called spike sorting. Using this pipeline, we would then seek to quantify both cellular and network level features of neuronal networks in order to infer comprehensive electrophysiological phenotypes. As I will outline later, we have started to develop such a pipeline as part of our first project. Finally, we pitched to make our pipeline and the data publicly available, ideally on the SDSC platform Renku. I will also talk about some preliminary steps that we took into that direction in the second part of my presentation. As I've introduced in a previous slide, in the first study, we used two cell lines of human dopaminergic neurons, one healthy and one engineered with a BT mutation. And we wanted to probe whether we can utilize HD MIA recordings for phenotypic screening. Philip Horner, a PhD student in our group, spearheaded this endeavor and spent most of this time last year to record the relevant data and to develop the analysis pipeline for this project. On this slide, you can see on the top left an example immunohistochemical staining of a healthy dopaminergic neuron culture. The dopaminergic cells are here depicted in green. They express trisimethylhydroxylase or TH. In the middle panel and on the right, we see a so-called electrical footprint of a single neuron recorded on a HD MIA and preprocessed using our pipeline. What I hope becomes apparent from this picture is the very high spatial resolution that we can achieve with our HD MIA recordings. This neuronal footprint allows us then to infer various features associated with the action potential waveform of single neurons, as the amplitude, its width, or several other metrics that could be potentially linked to aspects of ion channel dynamics and mechanisms involved in the generation of action potentials. In the present study, Philip inferred 17 different metrics at the cellular level, comprising both features that related to the activity of neurons and features that describe the waveform of the electrical footprint. The depicted statistics are based on features extracted from around 300,000 single neurons. Philip then followed these features over 5 weeks in vitro and compared how well individual cellular features would allow to classify our 2 cell lines. The lower plot, we see on the left a heat map that describes the relative difference between the 2 cell lines, wild type and A53T, and the accuracy results from a random forest classification analysis. We also highlight the development of 3 metrics. For example, the single cell regularity frequency on the top, which is a measure of the periodicity of the observed spiking signal, or the asymmetry, which is a waveform metric. In a similar way, we can now also look at the network level and infer features on that scale. Those relate more to the so-called network burst activity. Again, we can estimate a whole list of metrics, and we found in that study that the 2 lines actually differed quite strongly at this scale. In the lower plot, again, we see the heat map for the relative differences and the accuracy results that describes how well each feature would allow a distinction between both cell lines. In the next step now, we then use all cellular or network features or combinations thereof in a cluster analysis. The figure on the top panel shows all cultures in principal component space, and we see that both cell lines can be robustly classified belonging to either the wild type or the A53T group. So we have a robust electrophysiological phenotype here, both at the cellular and at the network level. The second plot indicates how well the 2 lines can be classified using either cellular or network features across development. You see high accuracy values, and again, the results look very promising. Finally, we also used the HDME metrics to ask whether we can predict the age of a culture and which features are the most informative for such a distinction. We find that age prediction is very accurate for the first weeks, and that there's a plateau after around 4 weeks, perhaps indicating a certain maturational state. Moreover, we found that there are specific waveform features that are more informative than others to predict age. Previous research has suggested that one strategy to target the underlying mechanisms involved in PD and to slow down its progression is to modify the levels of alpha-synuclein. And clearly, this is an idea that one could also test in vitro. In our study, we used an approach based on log nucleic acids in brief LNA to downregulate alpha-synuclein and to test the effect of LNA on electrophysiological properties. Although I will not go into details of this analysis here, our pipeline for electrophysiological phenotypic screening provided very interesting complementary results on what electrophysiological changes in both cell lines, and we hope to publish this in the next few months. A brief summary of the first part of my talk. First, I showed you that HDME recordings provide a wealth of detailed physiologically relevant features and allow for a systematic functional characterization of human neurons across development. I then gave a brief overview on our first study that found that we can use electrophysiological properties both at the cellular and network level to classify wild type and A53 human dopaminergic neurons and to study the effect of a compound that could potentially slow down the progression of PD. In the second part of my talk, I will present some preliminary results from 2 other projects that we are currently working on and that looked into the functional connectivity of in vitro neuronal networks. This is an aspect that we did not investigate in depth for our first project. Darren Kim, another PhD student from our group, probed whether the functional connectivity of iPSC-derived neuronal networks could be used to classify the aforementioned 2 cell lines. He therefore used the spike-sorted network activity of healthy and APP3 dopaminergic neurons, and then inferred the all-to-all functional connectivity between neurons using a range of publicly available methods. In the next step, he used graph kernels to train a support vector machine classifier in order to understand which connectivity inference method and which parameters were most discriminative. For the method study, found that transfer entropy, which is a model-free information theoretic measure, outperformed several other methods. Moreover, we found that the temporal window between which the firing of one neuron is assumed to be able to affect the firing of another neuron, so the postsynaptic neuron, here called embedding, is a crucial parameter that had an effect on the classification performance. However, what we did not address in the study, and this is something we will talk about in a minute, is whether the established functional connectivity also resembled the actual synaptic connectivity. We started to address this important question in the project led by Christian Donner, our collaborator at the SDSC. In a previous study, our lab has shown that HDMIA recordings can be combined with patch clamping to study simultaneously the extracellular and the intracellular of neurons and the input signal of a connected neuron to a patched cell. In the current study, we wanted to build upon this exciting approach to systematically test the synaptic connectivity of in vitro networks and to understand how accurate such connectivity can be inferred from the firing dynamics of neurons alone. As an initial step and to understand the requirements to reliably infer synaptic connectivity from combined HDMIA patch recordings, Christian came up with a phenomenological model to decompose the intracellular activity into synaptic contributions and a residual signal. In the schematic below, so at the bottom of this slide, we depict how the recorded extracellular spikes from putative presynaptic neurons are then picked up as a postsynaptic potential by the intracellular signal of the patched cell. Each postsynaptic potential is here colored depending on which cell it belongs to, so orange or brown or purple. Next, Christian simulated a network of 300 leaky integrated fire neurons with input from empirical spike trains and with known connectivity structure. So this was a model. In the panel on the left, you see a raster plot with some ongoing spontaneous activity, and below a simulated intracellular signal with either excitatory or inhibitory postsynaptic potential from 4 different presynaptic neurons. On the right, we see spike-triggered SE activity cutouts and averages for each of the selected 4 neurons and corresponding cross-productograms for each neuron pair. Since this was all based on a model, we can then run some performance tests and ask which parameters or recording conditions affect the inference performance. So for example, recording duration. As a next step, we will apply this framework to actual experimental HDMIA patch clamp data and thereby infer some empirical ground truth connections. Since we know from our HDMIA recordings where each presynaptic neuron and the patch cell are located, we can then ask how well synaptic connectivity can be estimated by their spike activity alone. We foresee that the inferred ground truth data will then allow benchmarking of different connectivity inference methods. The right panel show some preliminary results here for the inference of synaptic connectivity and some performance tests for excitatory and inhibitory connections. But this is preliminary data. As promised in the beginning, we are also planning to make this data and pipeline available to the community, and we have started to do so on Renku. As shown in this clip of our Renku repository, we'll have various tutorial scripts available that let the interested user see how connectivity can be inferred from spike trains. So here we're testing the inference algorithm with a cross-hologram based method online. So this is the result, the inferred graph. And then we can also perform some performance tests. Which would mean we compare the inferred graph to a ground truth that has been established by known connectivity. And this is, for example, the result. Okay, to summarize, in the study by Tim, we showed that functional connectivity inferred from human iPSC derived neurons allows to robustly classify healthy and diseased neuronal networks. And then in the last few slides, the project by Christian, I showed you some preliminary results from a modeling approach to test spike train inferred functional connectivity, and our attempt to establish ground truth that will allow to benchmark different connectivity inference methods. And then I also showed you our first attempt for a connectivity inference workbench on Renku called SpyCon. With that, I would like to conclude and thank everyone involved in this endeavor, particularly the members of the Bioengineering Laboratory and the members of the Machine Learning and Computational Biology Laboratory, as well as our other collaborators at the University of Basel, at Roche, and at the SDSC. Thank you very much for your attention.